Hello, my name is Nayeon Kim. I am a director in genomics division from Geninas in South Korea. I'm very happy to be able to present my talk and Twist Bioscience seminar today. Over the past few years, we have been developing and providing products and platforms for genome analysis for cancer patients in hospitals. In this period, we listened to the various needs from our clients and had a good experience with lots of challenges, which I would like to mainly talk about today. So I will briefly introduce our company for background and talk about status of clinical sequencing in Korea, challenges and overcome, and future directions. So Genius is a genome analysis company based in Korea. We make products that give optimal treatment selections by analyzing the patient's DNA or RNA. We are a sort of spin-off company from Samsung Medical Center in 2018. And before spin-off, we accumulated key technology within the hospital for over 5 years, including data generation, variant detection, various QC measurement for sequencing data, and new biomarker discovery technology. We have successfully launched Cancer Scan, a tissue-based NGS panel, and Liquid Scan, a liquid biopsy panel, and now we are providing those panels to major big hospitals in Korea. Recently, we have expanded our business to the single cell area, and we are carrying out several projects to discover various new biomarkers with pharmaceutical companies and hospitals. So this is not the new story here. Cancer is a genetic disease, and people are interested in various types of biomarker. Sometimes for diagnosis that can know the type of cancer or for recurrence of the disease. Biomarker is also an important indicator to know the response when a specific drug is used. There are a variety of known drugs and related genetic mutations, and this number is increasing, obviously. NGS-based clinical tests are becoming a standard test in many hospitals around the world because of the advantage of being able to detect these biomarkers at once at a very reasonable cost. In Korea, NGS-based tech is currently being actively used in both clinics and research field. In particular, insurance benefits began to be applied with the establishment of NGS clinical laboratory system in 2017. This is the status of the NGS-based solid tumor test in Korea. Last year, there are more than 10,000 tests, and the number will be increasing continuously. Most of the testing was performed in big general hospitals, which are major general hospital in Korea. And this is probably due to the complexity of NGS process. Fortunately, the system was quickly established, and so the patients are currently receiving the benefits of the NGS test. Although the clinical laboratory system in Korea has been stabilized, there are still many difficulties for NGS-based clinical tests to be performed smoothly in hospitals. The biggest reason is that the experimental process itself is relatively complicated compared to the other existing tests. After experiment processes such as analysis and interpretation follow, and for each process, it is necessary to decide whether to proceed or not through appropriate QC measurements. In case of patient samples from hospital, the amount of DNA is very, very small. Also, for storage purpose, you often extract DNA from FFPE samples, which results in some problems with your DNA quality. On the other hand, as new biomarkers are continuously added on the list of genes to be analyzed is different for each hospital. Customization of the panel is important, but the process of producing and validating the panel is also not easy every time. NGS result can give a lot of information at once, but it is difficult to store and utilize the information very well. Insufficient annotation can sometimes confuse interpretation. In Korea, NGS clinical tests are relatively inexpensive because parts of the total cost is covered by insurance, but it is still a burden for patients. So briefly, this figure shows the process of patients coming to the hospital for NGS setting. First, when a patient comes to the hospital, he or she consents to the test, and then test begins with a tissue biopsy in clinical practice. After DNA is extracted and library is created and sequencing is performed. Bioinformatics analysis is performed through various types of software, and when the results are selected and completed in the form of clinical reports, the report goes to the clinician. Our company initially did this kind of process within a hospital. And after spin-off, we have been supplying products in the form of DNA or RNA panel and software. We went through various trials and error to make this series of processes smooth. And there are three main parts that will be discussed today: customization of the panel and the quality of samples, especially FFPE sample analysis, and the automation of the NGS process through the system. Panel customization and optimization involves a variety of factors, including adding new genes or biomarkers to an existing panel, optimizing probes in the process, and planning a certain number of samples in one round within your turnaround time. Optimal cost should be determined, and validation should be standardized in the process. In the last 2 or 3 years, we have been able to work with Twist Bioscience to stabilize our products. I will quickly review about this first. We recently switched our entire lineup using Twist Bio panel, but today I will focus on the validation of our main panel, Cancer Scan and Health Scan. These are somatic and germline panels. We designed the target to the same level as our previous panel, and the number of probes of Twist Bioscience was significantly reduced, but the area we wanted to see was sufficiently covered. For panel validation, we basically conducted experiments with a single and multiplex. We pulled A samples for multiplexing and then downsampled them to specific read counts for comparison. In order to confirm the detection of germline and somatic variants, we used sample NA12878 that the correct answer is well known, and at the same time we used mixed material made by pulling 10 HAMMAP samples. We did some protocol optimization in consultation with Twist in the beginning, and it succeeded in raising the uniformity as shown in the figure here, which was similar to the single-plex experiment we were using with the existing panel. The proportion of reasons covered by less than 10x was very small. We investigated the coverage for the highly significant target variants. When Twist panel was used, it was confirmed that it was evenly distributed in the upper coverage. And this seems related to the high detection sensitivity, which will be mentioned later. We investigated the coverage for the highly significant target variants. When Twist Bio panel was used, it was confirmed that it was evenly distributed in the upper coverage. This seems related to the high detection sensitivity, which will be described later. When the average depth according to the GC content was investigated, the distribution looked similar with our previous panel. We checked a little bit more about the certain areas, especially the area that are not particularly well captured because of GC content. For example, the TOT promoter reason is one of the major reasons to be looked at in clinical practice, but because they have important variants in prognosis. It was confirmed that this region was well covered when designed as a Twist panel. We investigated the background noise that can be observed during multiplexing and confirmed that the results of Twist product showed relatively low noise, which was very satisfactory. We found that the sensitivity was higher than 95%, even when multiplexed. When we compared the allele frequency of the variants obtained by multiplexing with allele frequency of the correct answer set, it was confirmed that the concordance rate was also very high. I'm not going to talk about the performance of our panel itself here today. What I'm trying to say is that we found that the performance of the panels were improved by selecting a panel with high capture efficiency and optimizing the protocol, and by standardizing this kind of verification method, we were able to successfully upgrade all of our panel recently in a relatively short time. Next, we will talk about the specimen obtained at the hospital. In hospitals, actually various types of biopsy methods and the samples of various types of cancer are tested, and the amount of DNA that can be obtained varies. We have looked at the average amount of DNA by biopsy type and cancer type through a survey of about 2,000 samples. It is important for the clinical lab to establish such an internal guide by itself, and it is especially significant to make a guideline in consideration of the amount of DNA that can be obtained from FFPE. Since small amounts of DNA is inevitable in the hospitals, we optimized the protocol so that a library could be constructed effectively even when processing a very small amount of DNA. By optimizing the enzyme reaction conditions and adapter concentration, it was confirmed that the sequencing efficiency was improved more than 3 times compared to our existing method. FFP treatment of specimen is a sort of routine process in a pathology lab for storage, but there is a limit to the amount of DNA that can be obtained and the quality of data. We investigated the depth and uniformity of fresh and FFP samples for about 2,000 samples. As you can see, the uniformity and the depth concordance were relatively high in the fresh sample, but not in the FFPE. For example, although mean depth was very high, there were many samples with low uniformity. We thought that no single indicator could be an absolute QC measurement. One of the reasons is that the sample in clinical practice is very limited, and if you decide whether to report or not based on a specific single indicator, the number of failing samples will be quite a lot. To overcome this kind of point, we created the concept of PR score and made an index that can select samples that can be reported in clinical practice. That is, it lists the major variants to be seen in clinical practice and determines whether the variants have secured a certain level of depth. Using this method, even if the actual uniformity or depth is quite low, it is possible to identify a sample that covers major variation well and generate clinical reports. In the graph on the right, the samples in the purple circle are actually the samples with high uniformity and high depth, but very poor PR scores, which cannot be reported in the hospital. Quality issues caused by FAPE also leads to other kinds of problem, one of which is many false positive variants. For example, when detecting structure variation, this effect is much greater, and many split reads occur due to the DNA fragmentation and some sort of modification, which result in false variant calls. To overcome this problem, we developed our own caller that can detect fusion of FP DNA well, and the key idea here is to calculate the diversity of split reads, filter them, and select the best candidates based on the local assembly. It shows higher specificity than the current public callers, variant callers. The last thing I would like to mention is automating the system in hospital. As mentioned briefly earlier, since the process of NGS test is complex and generated data is quite huge, it is very important to establish a process at each stage and use appropriate software and platform. Since this issue is directly related to the turnaround time, it is particularly important part of hospital setting. We are overcoming this by developing the platform called OncoStation, mainly when an order comes in from the clinic through the hospital EMR system, electronic medical record system. The clinical information goes to the OncoStation, and all NGS processes are managed on this platform. This is the entire QC process in the experimental process and bioinformatics analysis process from FASTQ to VCF after sequencing, all managed on the platform. Final results are transferred to EMR as well. In this process, we support clinical interpretation by using various types of in-house knowledge base. In other words, we provide not only annotations for targeted and immunotherapy, but also clinical trials information and the frequency of the variants based on the data we have been accumulating. We provide a total solution required for NGS operation by providing various levels of statistical information, such as test status and some sort of sample information and mutation status. In particular, we are looking forward to the active expansion of liquid biopsy, and we started to provide a real-time follow-up system for each patient. So, for example, if the same patient performs genomic analysis through a tissue or a liquid biopsy, the clinical results can be followed up as shown in this video. These efforts are designed to make it possible to easily access and manage more data in clinic and pathology lab. And these are actually the needs in the clinical field. Okay, this will be my final slide today. NGS-based clinical tests are currently being used in many hospitals over the world, and its role is expected to rapidly increase. So data from tissue or liquid biopsy will continue to accumulate, which will be combined with clinical information to create secondary value. We believe that the platform like OncoStation will play a very, very important role. And based on this drug target discovery of new biomarkers, or even like AI-based prediction, something will be possibly developed based on this kind of databases. We want to make this innovation possible through collaboration with various experts, including pharmaceutical companies or the major big hospitals. So thank you very much for your attention, and I'm very happy to answer any questions you have, or you can just email me anytime, and thank you very much.